Hi, everyone. Welcome. My name is Scarlett Bliss. I'm a senior at Tufts, and you've probably met me at least virtually or by email at this point. Thank you so much for joining. I'm just going to be accepting participants here we go, as they come in. Forgive me for that. But so I'm so excited you're all here and thinking about and planning to teach PARE-Seq this spring. We're really excited to support you in that process. I'm going to give a big overview of the course, what you can hope your students would get out of it, the course schedule, as well as a brief overview of Galaxy as a platform, which is what we're running the bioinformatics activity through, how to teach that to your students, and then there'll be plenty of time for questions, hopefully at the end. If you have any questions throughout, please feel free to type them in the chat or to raise your hand, and I'll try and monitor that as I'm going, or just jump in if there's free time. All right, so we'll get started. So I'm just going to start by introducing everyone that's part of the PARE-Seq program. So of course, you all know Dr. Carol Bascom-Slack, so Carol's on as well. Carol started PARE, and I've been working with her and Dr. Amy Pickering, who is a professor who was at Tufts for many years and is now just transitioned to working at UC Berkeley. And so the two of them I've been working with since for the past few years with the PARE program, along with Erica Furmeister, who's the postdoctoral fellow in the Pickering lab and has been incredibly helpful in this whole process. And I'm a biology and community health major at Tufts, who's been working with the Pickering lab for the past few years. And I'd like you to introduce my co-teacher, Eve Abraha, who can take a second and say hello to you as well.
Hi, everyone. I'm also a senior at Tufts, like Scarlett, studying biology and education. And I'm also one of the co-teachers that y'all have probably seen our Seek videos.
Yeah, so Eve is awesome. And we kind of split out the teaching and are both working on kind of surveys and assessment that are involved in the pre and post survey as well to assess the efficacy of the teaching mechanisms that we're using in the course. So I'm going to begin by just giving an overview of the website. I hope that you've all had a chance to see it, but just give you a tour of where everything is, and then we'll go over a general structure of the course. This is going to bring us to the PARE-Seq website, so you can find it at www.pareseq.com. And when you get to the home page, you're going to see this pop up on the side. I believe it's still loading. Here we go, of the pre-course survey. So I say this later on, and I'll discuss the evaluation surveys later on, but we encourage all of you to discuss with your students taking the pre-survey before you begin the course, and then the post-survey following. That really helps us get an idea of how the course has helped your students learning, and kind of engage with the material. And hope to improve it in the future. So there's an introduction video right here. Even I have just been working on recreating that introduction video, so a new version will be up in the coming few days, with a little bit more information, a kind of more conversational version with the two of us discussing our experiences as students and kind of the importance of, or the significance of, us teaching this course through kind of a peer learning framework. So us being students and hopefully teaching students, and trying to be as approachable as possible in this whole process, because personally I think that learning from peers is a really kind of effective tool to feeling engaged and confident in learning STEM. So when you scroll down, this is just a scrolling bar of all of the video lectures, but they're also present in the correct order when you go a little bit further down into this home page. This is a paragraph explaining the course schedule, and we give you a potential breakdown into 2 or 3 parts of this course, but please feel free to use the videos and the material however it works best with your classes. We know that everyone is teaching in different formats this semester, and we've broken it down into short videos that are hopefully digestible and able to be taught at different times, to make this as flexible as possible for you all. So part 1, and I'm going to go over this in greater detail in just a minute, offers a background on antimicrobial resistance, and that's 4 videos here. Part 2 is applying that understanding of AMR to learning about ARG, antibiotic resistance gene detection, through using bioinformatics, and this open source online platform, Galaxy, which then students have a video tutorial right here that they can use to complete the Galaxy activity. And then we have two options for final projects, should you choose to teach them with your students, either a final lab report format or a final poster presentation format. So that gives you a little bit of flexibility depending on what works best for your class. This is a little bit about us as a group. And then up here, you can go onto the materials page. And this is where you'll find all the information, and you can direct your students as well to find the information for the course. The two final assignment pages for the poster presentation and the lab report are housed here, as well as a written Galaxy instruction manual. So should you be a more visual learner, and just like reading along the steps when you're conducting the Galaxy activity, that's where you can find that information. And then also for all of you, there's a detailed instructor packet right here that will provide a bit more of this information that I'm providing you right now, as well as links to an additional whole set of sequence data if you want to give a variety of sequence data to your students. So not just one single soil and water sample as it's given in the Galaxy instruction manual, but if you'd like to give different samples, quote unquote samples, to your different students for the soil and water sample for the Galaxy activity, that is all found in the instructor packet. And then finally on this site will be the evaluation surveys. So this is the pre and the post survey for your students. It should take max 10 minutes. And it is composed of both knowledge questions, so a set of the same questions before and after to assess learning gains, and then a set of kind of self-efficacy questions and confidence in the material and STEM courses. And so it's all anonymized, and none of it will be used obviously for grading. And for that reason, I've talked with a few of you this semester. We can't give you the specific results for your students with their connecting to their contact information for IRB privacy purposes, but we can give you aggregate results for your classes if you're interested in seeing kind of student results from those surveys post teaching PARE-Seq. And then for all of you instructors, after you teach, we're just really interested in getting as much feedback as possible to keep improving the course. So there's also a post-course instructor evaluation survey right here. And we're now going to go through all of those in a bit more detail. I just wanted to show you where everything is housed on the site. So part one, as I mentioned, is this background on antimicrobial resistance, and it contains 4 videos. The first video is called Understanding Antibiotics and Antibiotic Resistance. This video, the first 3 videos are taught by Eve, and then the second 3 videos in the whole section are taught by me. So this is going to give all students a background on antibiotics, antibiotic resistance, and giving that fundamental knowledge that generally a student will learn in a biology course. That sets them up to take video lecture 2, Antimicrobial Resistance Spread Through the Environment into Our Bodies, which hopefully will give that context of how the environment is a transmission pathway for antimicrobial resistance, and thinking about this issue through a One Health lens, the idea that the health of humans, animals, and the environment are all inextricably linked, and you have to understand this pathway of AMR through paying attention to all 3 of these transmission pathways. Third, we put this into context as a huge public health, a global public health issue. And this video is going to include an interview with Dr. Maya Nadampally. We'll talk about the guest interviews in just a minute. And so we talk about AMR and how it disproportionately impacts many traditionally marginalized communities and low-income countries. And then we finally discuss 5 specific families in lecture 4 of antimicrobial resistance genes that we're going to focus on in the Galaxy activity, so in the application part of this course. And the 2 learning goals for this section are listed here. We wanted, especially this semester, we've been working a lot on kind of the curriculum development over the fall and making very explicit learning goals for instructors to be kind of aware of what they hope their students are going to get out of it. And then also for students to make sure that they can check in with these learning goals and be sure that they can explain these different concepts as they move through the course. Any questions before I move on to part 2? All right, so part two is the application component of this short course. There are 3 videos, 2 lecture videos, and then a tutorial for doing the Galaxy activity. So we run through, we've done these, made the biggest changes to these videos if you haven't seen them since the beginning of last semester, because we've really spent a lot of time on breaking down the ideas of sequencing and bioinformatics and making sure that we're teaching them as comprehensively as possible to the students. So in the tools of bioinformatics class, we are going over basically all the different processes that you can do to identify antibiotic resistance genes. So we cover culture-based approaches, we cover PCR-based approaches, we cover what sequencing is, and then what next generation sequencing, or specifically nanopore, which is the type of sequencing that we're using in this course, is, and why we're choosing to use nanopore sequencing. I'm going to accept another member. And so this is a really helpful video for students in understanding where the data for this activity comes from, and kind of explaining where you get that FASTA file of A's, T's, G's, and C's that then you're going to be able to find what antibiotic resistance genes are present and specifically what species they originated from. Then we go on into video 6, which is specifically doing a deep dive into what Galaxy is, what a workflow is in bioinformatics, and how we've built this specific workflow of tools that will take a FASTA file, a sequence of water or soil metagenomes, and the metagenomic data from those samples, and figure out what species are present in that sample, what antibiotic resistance genes are present in that sample, and then what species those resistance genes originated in. And so we explain all the different tools in that workflow, and then kind of basically set students off into running Galaxy themselves, hopefully understanding kind of what that process is doing for them. And you can see those learning goals assigned right here. So I'm just going to run through this activity workflow really briefly to make sure that everyone understands. Please jump in if you have questions, but this is kind of the process that we're using from start to finish, that students are involved in kind of the back half of. And the overall goal is, so we're providing these FASTA files of sequence data for your students. In an ideal world, we would have a nanopore MinION like sequencing technology available to all of you. So you and your students could be going out into your environments, collecting soil and water samples, extracting your own DNA, and then running this Galaxy workflow to detect antibiotic resistance genes. And just like the original PARE module, that can be a really awesome environmental surveillance tool to be able to map kind of hotspots of emerging resistance nationally. So that's where we're heading in the long term. Would be great if we can turn this module into that type of environmental surveillance tool. For now, though, we've collected, the Pickering Lab Group has collected the soil and water samples. They come from samples from India and Kenya, and then they've been subsetted to ensure that students are going to detect the resistance genes that we are studying in this course, so the 5 families that I discussed in video lecture 4. So the DNA has been isolated from those samples. And then we've extracted all of the metagenomes from all the biological material from those samples and sequenced it all using a nanopore MinION. So this is a really cool tool run by this company, Nanopore. That's a next generation, third generation metagenomic sequencing tool. It's about the size of a flash drive if you're not familiar with it. And we discussed this in video lecture 5. And you can input your sequence data, plug it right into your computer, and get these FASTA files from it with all of the sequences of the metagenome. So by using these FASTA files that we're then going to give students, by running them through the Galaxy workflow, they can identify the antibiotic resistance genes present and then assign them to their host species. And so that is what the Galaxy activity that students are going to be completing will be achieving. And the database that we've curated that basically they're doing an alignment to from this FASTA file just includes the sequences for resistance genes of the 5 families that we've discussed. So those are the TET family, MCR1, colistin resistance, NDM1, carbapenem resistance, ARM-A, and I'm forgetting one, CTX-M15, blah. So a group of beta-lactamase resistance genes. So along the way, we also have a number of guest interviews of kind of quote unquote experts in the field of AMR. And we're hoping that these will contextualize for students ways that they can get involved in research or clinically in the work around antimicrobial resistance detection and management. So Amy Pickering is one of my PIs who now works at UC Berkeley, but has worked at Tufts for a number of years. She'll provide an interview that goes into detail on what it's like to be a researcher. So on the kind of laboratory side of research around AMR, the importance of environmental stewardship for AMR and environmental surveillance, and then also how students can get involved in what a professional, so a person in her position, looks for in a student who's interested in getting involved in research. Dr. James Kirby, and this video is not on the website yet, but it's being edited now and will be posted shortly, is a Harvard medical faculty physician, and he's been really involved in the PARE project from the beginning and is really helpful in helping us determine what resistance genes to study with students that are of specific clinical significance. And he gave a really great expert opinion, kind of as a medical professional, on what it's like to be involved in the clinical research for supporting patients in supporting hospitals in the capacity of helping patients figure out what antibiotics they should be using for specific bacterial infections. So we're hoping that this video will help students who are on the pre-health tracks taking this course understand the importance of antimicrobial stewardship as medical professionals and see how that role is crucial to their future careers. And then finally, oh my goodness, it says Amy Pickering. This is Dr. Maya Not-Impali, my apologies. Dr. Maya Not-Impali is a research assistant professor in the Pickering lab as well at Tufts, and she's really active in CMAR, the Center for Integrated Management of Antimicrobial Resistance, as well in Boston. And she's going to talk about the disproportionate impacts of AMR on low-income countries and traditionally marginalized communities, and why it's important to be working on this work kind of from a public health standpoint. So now, unless there are any questions, I'm going to give a quick overview of the Galaxy platform. For those of you who have been on one of these webinars before, it will be pretty similar to the Galaxy tutorial, but I'll keep it brief. I just want to overview for you all what Galaxy looks like and basically use this time to instruct you really quickly on how to use it. It's a very user-friendly platform. And we've built the workflow for your students. So mostly what they're going to be doing is entering their data. All right, hold on, I've lost my mouse. Apologies, just 1 second. Okay. So when you get to Galaxy, the Galaxy tutorial video will instruct students on how to make an account. It's free, all it requires is an email. And once you get to Galaxy, this will be the home page. You'll notice that we're using Galaxy Europe, and I've explained this before, but basically the only reason for that, there's a couple different servers for the Galaxy platform. There's a galaxy.org, galaxy.uk, galaxy.eu. We use this one because of the time difference. There's generally a lower load on the international servers at the times that students would be using them in the US. And so it's just a little hack around to make the workflow run a little bit quicker. But all of the platforms are the same, the European and the American versions. So once students reach this website, they're going to be instructed through the tutorial and the written instructions to import their data over here. You're going to import the FASTA file links, which are found in the instruction manual right here when you click paste or fetch data, and then you just paste the link to the FASTA file right there. So all of the links to the FASTA files are uploaded to a website called Zenodo, which is kind of like Dropbox or Google Drive for data. And so just by pasting in the files, that will start the upload process. They also upload the database that we use, the specific ARG database that we use for this course. Then they're going to go to the workflow button and go to shared data. They can click on workflows, and they can search the specific workflow that we have published for PARE by just searching PARE-Seq. So there you'll see the PARE-Seq ARG's workflow, and by clicking import, they can import it to their own account. So once they've imported it, if they click on workflow, these are where all their imported workflows are, and you see it right at the top. And if you click the run button, the workflow has been uploaded for them with the specific set of tools that allow them to get the specific outputs that we're going to discuss and are analyzing, and help them find the specific information that we're going through by putting in the correct files, which we instruct them to as well, the water file or the soil file, and then the reference sequence, which is the databases that we've built with the specific ARGs we're studying. They can just click run workflow, and it's going to send it right into this side histories window where you'll see the boxes turn orange when they're running, and then turn green as soon as the tasks have been completed. So this is all of the data for a specific water sample number 5. And once it's done, it'll turn green. And you can see once by clicking on the eye symbol next to any one of these, you can see the outputs. So the key outputs we're looking at, which we also go through in the courses, are this summary statistics report, which gives some general features of the data. And then an output of the taxonomy present in the sample. So this is a list of all of the species present. You'll see a lot of them are unclassified, but a lot of them are classified. And this is a very long list. And then a pie chart, which gives a much more interactive way to analyze all of the taxa present in the sample, which you can zoom into to find the most abundant species, for example, by clicking on the largest portion of the pie repeatedly. Then you can go to the alignment table, which is mini map 2. This is going to give you all of the antibiotic resistance genes present in the sample. So in this sample, you can see that there are a number of different tetracycline resistance genes as well as CTXM15. And then finally, kind of the crux of this analysis is this ARG taxonomy table where they can see the species present and then, sorry, the ARGs present and then the species which they came from. So this is really helpful in understanding what bacterial species are harboring resistance genes in this sample. So these tables can be exported or just copied and pasted into an Excel or a Google Sheets list. And we explain that whole process and then including these final key outputs in the lab report or poster presentation that they then produce for the course. And that's where we'll get to the final project. I'm going to be posting pretty soon, and we have basically assignment sheets for both of these options should you choose to use them with your students. But we're hoping to also post sample lab reports and poster presentations, and work with any of you interested who have rubrics for a project like this to develop a sample rubric for a poster and a lab report. So students are is very explicit what is to be expected of them. But of course, you as the instructor will be grading or choosing what type of final project your students will be completing for the course. And then in terms of the evaluation surveys, like I mentioned before, the pre and the post survey are found on the PARE-Seq website. And we're really hoping to assess the learning gains from the course, as well as any potential interests in careers or careers in STEM or research in STEM that they've gained from taking this course. And finally, the impacts of peer learning, of diverse representation in teaching staff, and of teaching and from learning in this format that's not exactly test-based, it's more project-based and provides a diverse set of learning tools to understand the material, and whether that's impactful for learning a specific skill set, such as bioinformatics in a virtual setting. And so finally, I just wanted to provide these information. Just please let me know if you have any questions or technical difficulties throughout. I'm very willing to do kind of one-on-one meetings and go through Galaxy at greater detail or anything else should you have questions. I also joined a couple courses virtually through Zoom this past fall, and it was great to meet some of your students. So I really am happy to do that if you'd like to include us in that process in your teaching of PARE-Seq. Please encourage your students to complete the pre and post surveys. And then also I just wanted to let everyone know that Eve and I are working on adding resource pages for each topic, so for each video to this class. So other additional YouTube videos or websites that provide information, either if students would like to look into any particular parts of this issue in greater detail, or if they've had any confusion from learning the classes. So this will just provide more information and ways for students to engage with the material. And so, yeah, I'll leave it at that. If anyone has questions, I'll just open the floor to anyone else.
Scarlett, there's a question about
Yes, sorry, I can't see the chat. Let me get the chat up.
Hi, Scarlett. This is Donna Sue, and that's
Hi,
my
Donna.
question. So, and we also, I feel like we had I don't know, one thing, we did this at the end of the semester, which was, good because we were able to sort of incorporate a lot of what we had done into the semester, into the assessment. We sort of modified the assessment a little bit, but bad because our minds were all so bamboozled by then that, everybody's hanging on by a thread, I think. So one of the things our students had some, not a large population, but a few of them, of course, the most aggressive bulldog students, was that there's a soil sample and a water sample. Mm-hmm. And they really started focusing in on this and saying, well, are there, does antibiotic resistance, is it different between water and soil? And what I kept trying to tell them is as per our conversation, was to focus on the geographic location. But so there's two questions to that. Is there a reason why one sample is soil and one sample is water? Because it introduced this confounding variable to them that there was two different media that the sample came from. And the second question is, I know in the assessment it said, so what difference is, what is the relevance of this coming from Kenya versus India? But is there anywhere when you're putting in the data that says this sample is from Kenya and this sample is from India, or is it only, and I could miss it someplace along the line. So it's just the one thing that kind of really ended up getting hashed out by our students, because once one of them grabbed onto it, they were all talking about this. And so listen, I'll say all, meaning the 4 or 5 who contacted with me. I wasn't responsible for the classes as a whole, so.
Absolutely. And I'm going to let Erica contribute as well. But what I'd say at first is we've really intentionally not included their locations, that they specifically, these samples they came originally from India and Kenya from research that was being done in those locations. But we've manipulated those DNA sequences to make sure that we're including the specific ARGs of interest to this course. So they are fabricated in some sense, these samples. And so that's why we've intentionally not included their locations, because we didn't want to assign the kind of incorrect assumption that, oh, okay, I'm finding just tetracycline resistance from this water sample from XYZ country, and then I'm going to look into that specifically. These really are kind of hypothetical FASTA sequences, and then the hope one day is to then be able to use real samples from environments.
So that question in the assessment about Kenya versus India is,
I don't think that there is a question in the assessment. I don't know if you created a different assessment, but to my knowledge, there is no mention.
Where did I come up with that then? Is it in the instructions? Is it someplace there's something about Kenya versus India?
That might have been a conversation we had on the side where I was explaining where the samples originated, but I don't believe we included that in the material, just because we didn't want to create that kind of false.
Okay, I'm gonna take a look as we talk, but okay.
I can also provide a little more to this answer. The reason that it's soil and water is basically because that's what we have in our lab right now. This semester and through the summer, we're going to be sequencing more soil samples so that they could potentially be included in this PARE-Seq data set. But basically, we used what we had. And as far as not wanting to provide the exact location, I think we're trying to be culturally sensitive. Okay. Like, we don't want to imply because we purposely picked samples that are going to be interesting for the students, that's representative of like this India and Kenya location.
Okay. But there's no reason, is there a reason why we don't have 2 soil samples or 2 water samples? Because that was
we don't have like nanopore sequencing data from our lab from 2 water and 2 soil.
Okay. I'm opening up the lab report now. I do not so where did I come up with the I don't, it is in it absolutely is. It's in number 4 in the discussion of the assessment for the lab report. It says, what is their clinical significance? How is this relevant in the context of their origin, Kenya and India respectively?
In the lab report assignment?
Yeah.
I'll go back and check that through. I don't think that that meant necessarily like Kenya and India respectively for their locations, but just in general, the context of,
Okay.
I think that if anything, that was pointing towards understanding that that antimicrobial resistance is a larger public health issue in many low and middle income countries. But I'll definitely address it. Thank you.
And the reason I ask this is because I remember this, you know, this student who had grabbed onto the soil versus water, and I had a conversation with you about it, Scarlett, which was, is this relevant that it's soil versus water? You know, I don't know. And from what I remember, your response to me was, no, the geographic location is really what is more important than whether it's soil or water. So then I tried to sort of shift them to that, but I guess then as an instructor, I'm confused as to how they're supposed to be answering, I guess not exactly how they're supposed to be answering that question, but what is the learning goal associated with that question in the assessment?
Yeah, so definitely, I don't think that it's necessarily a learning goal associated with the question, but I'll go back and look at the lab report assignment.
Okay.
And in general, in terms of the soil and water question, we talk a lot about kind of the transmission pathways of antimicrobial resistance through the environment and where it can be released. So for example, a pharmaceutical company dumping into effluent would then, you know, impact the antibiotic resistance genes potentially in water samples in comparison to soil samples that might be detecting antimicrobial resistance genes near CAFOs or agricultural areas. So the resistance genes might be occurring and basically the resistance might be transmitting through soil versus water samples from different sources. But other than that, there wouldn't be kind of a difference.
Okay.
We just wanted to provide multiple different mediums of environmental DNA.
All right, thanks. It's super helpful. We used, you know, the assessments were really good, and the questions you asked again lended themselves towards us sort of bringing some of the concepts they were learning in PARE back to things we had done early in the semester. So it was a great summative assessment for our students at the end of the semester.
Great, I'm glad. Thanks, Anna.
Yeah.
If I could just jump in for a second, you bring up really good points, and I do think that the area, Donacy, where you were talking about where the question was about origin, is that the lab report questions or those questions were written early on, and you all know, you know, things evolve, and we're now working on the second iteration. So we'll definitely go back and look at those. They may not be, they may need to be rewritten. But the other thing I want to say is I just want to give a big thanks to the many PARE instructors who piloted this fall. We always appreciate people taking time to pilot something that's new and give it a trial run for us and give us feedback. And so we encourage you to continue to give us feedback. And, you know, as you've brought up, perhaps we need to think about how we package the DNA samples, the FASTA files, how we kind of advertise them. But as with everything PARE, this is one step along a longer trajectory where we're hoping that eventually we'll be able to give bigger, better, more variety of samples. So it's always a work in progress, and we really appreciate any feedback that can help to make it better, and, you know, things that are not working for you. It's really helpful to know.
Any other questions while I'm on?
I have a quick question. First of all, it's been really awesome to watch the evolution of PARE-Seq from the summer when we first started talking through now. So awesome job, and thank you for including me in it. My question has to do with the evaluations. So my class wasn't able to participate in them last semester because they weren't available when I was implementing PARE-Seq. But I have two different classes where I could see using some of the videos. I have a public health class where it's a gen ed. We're not really getting into bioinformatics, but I think that your initial videos are really, really useful. My question would be, would you want those students to take both the pre and post take no evaluation, kind of where would they fall?
Yeah, I mean, I would definitely say I think they should still include them. What you can do is we ask a question selecting which parts of the whole module the students have participated in. So if we see when we are going through the data analysis, if we see that they've just done those introductory courses, or especially just the video on the public health implications, we'll know not to include those in the knowledge question component, but it'll still be really interesting for us to see the answers to those kind of like career interests and self-efficacy questions.
Perfect. Great. Thank you.
Yeah. Thank you, Katrina. Any other questions?